to allow some passage, right? Some are going to get through easier than others. Okay? Easy passage across the membrane. This is going to be simple diffusion for the most part.
It's like 9 questions.
Small, non-polar, hydrophobic molecules. And when we say hydrocarbons here, again, it has to be small hydrocarbons. It can't be one that's, you know, a meter long or something. Well, it's not a meter long. But anyway, it can't be the super large hydrocarbon, right? Small hydrocarbons. Carbon dioxide, oxygen, nitrogen. These diffuse across the membrane directly by simple diffusion. So CO2, oxygen, nitrogen. Non-polar. Gonna diffuse easily. Cool. Difficult passage or protein-assisted passage. This is going to be for hydrophilic molecules. Why do you got 2 or 3? So you can alter them, huh?
No, it's for my teammates. Because I'm a good teammate like that.
You are, yeah. What do you got in here, Dina?
Well, on Aries I have the D, but on McKinnon's I have C minus. So it depends on who you're asking. Wait, don't do the same pen. You're gonna get mad at me. Don't put Scarlet sucks. Okay.
So, hydrophilic, polar molecules, large molecules, ions. These are going to need help getting across the membrane. Let's see, Zarifa. Shad. Collect. Students walking to local elementary around me and others. 10:15. Okay, yeah, you guys are going to a elementary school? Yeah. Very good.
You're going to elementary school?
Don't be scared of the damn kids, dude.
I'd feel bad. They're elementary. You know what I did in elementary? I was a devil.
1 point each?
Yeah. On the multiple guess and the matching 1 point each.
This is Billy.
32.
Precious has a little but everything else is identical. All the other questions are identical. So, and water. Here's something that's interesting is usually you guys are taught that water diffuses across the membrane because we talk about osmosis and water just diffusing across the membrane. What we don't tell you until you get to this level, and I have no idea why, because I think you can handle this information. I don't think it's like rocket science that, oh my gosh, can't handle that. There's a specific channel that water has to go through to easily go in and out of the cell. And that's a channel called an aquaporin. That's millions of water molecules in and out. per second. They're called aquaporin, but, and it's a specific channel just for water. Two main types of transport, passive and active. Right? This should be really familiar from ninth grade. Passive transport, active transport. Passive means what?
To allow?
What's a pacifist?
Oh.
Nobody knows what a pacifist is?
Isn't that like a happy person?
Happy, maybe not, I don't know. What's that? Anyone? Anyone? Somebody who does not fight. Right? They don't fight
That's the same.
necessarily.
Passives and ultimates.
Right? Oh, what does a pacifier do for a baby?
Feel like, so let's fight and be like, no.
It soothes them.
pass-
Both those instances, somebody not fighting and a baby calming down decreases the amount of what being spent?
Energy?
Energy, right? If you're not spending energy to fight, because you're a pacifist, right? You're not spending energy to cry because you're pacified with a pacifier, right? That's why we name it that, right? Decrease in energy. So, passive transport. Transport that does not require energy. Cool. Active. If you're active, you use more energy, correct? Active transport. Transport which requires, right? So it moves to down the concentration gradient. I'm trying to put as many examples, ideas, phrases into your head as I possibly can to cement this idea. Where it's going to get goofy is when we start talking about osmosis. Hypertonic, hypotonic, isotonic, you're gonna goof those terms up. I'm telling you now, you're going to goof them up. So you need to get as squared away right now as you can with concentration gradients, electrochemical gradient, up and down gradients, all, right, concentrations, all that you need to get as comfortable as you can right now, so that when we get to osmosis, you don't get messed up. Passive transport. We have a high collection here, a low collection here. Extracellular cytoplasm. The yellow diamonds, where are they highly concentrated?
At the top.
At the top, so in the extracellular fluid, right? Up here. So they are going to passively diffuse through here. No energy is going to be required. And they're going to try and reach a state of equilibrium. An important thing to understand, you do not ever want to reach a state of equilibrium in your cells. If you do, you're probably going to die. Because that means nothing is moving anymore. Right? When we talk about things moving back and forth, we can have it's not apparent. What's the word? Something equilibrium. Crap. I just lost the word, dude. Dynamic equilibrium. And that's where, okay, things are still moving. It appears that it's in equilibrium because there's no net movement of molecules. Molecules moving back and forth across the membrane at the same rate. 6 go in, 6 go out. 6 go in, 6 go out. That's dynamic equilibrium. There's no net movement of molecules. That's fine. We can get to that. But if you reach true equilibrium, you're going to die. Molecules diffuse directly across the membrane, different rates of diffusion for different molecules. Yeah, and the membrane is selectively permeable. Let's throw this one at you. If I had to ask you to describe these molecules, these greenish yellow ones, what are terms you could use to describe those molecules?
Polar.
Are they polar or are they non-polar?
Polar.
Are you asking or are you telling me?
They're polar.
Okay.
Because they're having difficulty diffusing. They're slower than the other one. I think. Maybe?
So let's look at, again, passive transport in your notes 2 pages ago, 1 or 2 pages ago. What types of molecules can passively transport directly across the membrane? So, what types of molecules pass through the membrane directly? Small, that's a good descriptor. Small, hydrocarbons. Examples would be carbon dioxide, oxygen, nitrogen. Are they hydrophobic or hydrophilic? Hydrophobic. Does that mean they are polar or non-polar?
Non-polar.
Non-polar. There. That's all the stuff you should be able to put together. If it's like, hey, describe the molecule, that can, any of those describe molecules that can pass the membrane easily, correct? Small, hydrocarbon, non-polar, hydrophobic. Examples would be carbon dioxide, oxygen, nitrogen. Right? That needs to just flow, dude. Are we good? Those are fair questions. You're watching cells, and you're watching cells under a microscope, and you place a radioactive tracer on a molecule, and you see it diffusing rapidly into and out of the cell. What could you conclude about that molecule? Alright, it's diffusing rapidly into and out of the cell. It's probably not going through an ion channel or anything like that. So it's probably small, hydrophobic, non-polar, hydrocarbon. It could be any of these things, right? That is a fair question, dude. Super good. Alright. Passive transport, again, this is a selectively permeable membrane. We call this, when we set these up, and you probably will see these in an assessment, this setup is called, anyone guess what it's called?
Passive transport?
No, this system here, this tube. Any idea?
What's it look like?
Okay, what do you suppose it is?
A U-tube?
Yes, absolutely. This is the original YouTube.
Oh my
That's
god.
that trash on the interweb. So this is a YouTube setup, right? We have a tube, open on both ends, a semi-permeable membrane in the middle. We put water, a solution in there, right? A solute solvent concentration, and we can watch what happens to the level of the water. Here we have sucrose molecules. There's 4 sucrose molecules over here, right? We're not going to like put 4 sucrose, anyway. Sucrose cannot permeate the membrane. Water can permeate the membrane, or the membrane is permeable to water, is another way to say it. So if we have this concentration, the water is at the same level, we have a higher concentration of sucrose molecules here. We have a lower concentration of sucrose molecules here. After a certain amount of time goes by, we look at it again, the water level is here and here. Sucrose molecules. What has happened and why? What has happened and why? Alright, let's just tackle the first part. What's happened? This is time A, this is time B. What happened?
The water went to the right more.
The water went to the right more. Okay, so the water permeated through the membrane. Right? Did the sucrose molecules?
No.
No, because the sucrose can't, the membrane is non-permeable to the sucrose. Okay. Wait a minute. Was any energy involved? Do you see any input of energy? Any little starburst that says ATP or anything like that? No. So no energy was input. So what process happened? A form of passive transport, which form?
Concentration.
Well, we got two answers.
Osmosis.
Osmosis.
Oh.
Usually, the first answer we get is diffusion. And then I'm like, yes, diffusion. Diffusion of what? Diffusion of water. What do we call that? Osmosis. But wait, Mr. White, you said osmosis and diffusion is movement of higher concentration to lower concentration. But the sucrose is more concentrated here. Did the sucrose move from high concentration to low concentration?
No.
No. Does that make sense?
Yeah.
This is where the little mind twist has to happen. If I have a high concentration of sucrose here compared to here, what can you tell me about the water concentration relative to each other on both sides of that permeable membrane? If I have a lot of sucrose and a little sucrose, what does that tell you about the water concentration?
It has to balance out.
No. You do a lot of whispering.
John.
Yes, absolutely. They are inverted, right? This is where kids goof it up. The relations are inverted to one another. If I have a high sucrose concentration, that means I have to have a low water concentration. And this is where things get mixed. When we start asking you guys, sometimes we'll say, oh, this relative to this. And is the solution hydrotonic? Or is it hypotonic? Right? This is where things are going to start getting. So if I have a high sucrose concentration, I have to have a low water concentration. Right? Because I have more sucrose molecules. They're the same level, right? These guys are sitting at the same level. I have 200 milliliters on each side. Let's just say that, right? But I have a bunch of sugar on this side and not on this side. Well, what does that mean? That means I have less water. I'm still at the 200 milliliter mark. More of the space is being taken up by the sucrose molecules, which means I have to have less water molecules. Does that make sense? And then the water, because the water is what moves, the water moved from where it was more highly concentrated to where it was less concentrated. There's less concentration of water here because there's more parts of sugar. Another way we can look at it, so if you can put it in your mind this way, so I've got my tube, and in my tube I have 1,000 pieces, or I can fit 1,000 molecules on each side at this line, right? And what I'm going to tell you is on this side I have 750 molecules are sugar. Well, what does that mean? That means 250 are water, right? Out of here, if I only have 300 that are sugar, that means I have 700 molecules that are water, right? What is going to happen? And this is only permeable to water. Water can go through, the sugar can't. What is going to happen?
The water's gonna permeate through the right, to the right.
Yep, so if this is side A and this is side B, water will flow from A to B. Until when? Or to what purpose? How much? If I have 700 and 250, how many total water molecules do I have? I have 700 water molecules here, 250 here. How many total water molecules do I have?
950.
950. What's half of 950?
425.
Almost. 450 is half of 900, and then half of the 50 is 475. Water is gonna go until there's 475 water molecules here, 475 water molecules here. That's what is going to happen until they get to balancing. And then what's gonna happen is, all right, so getting more in depth into this is now we would have to look at the percentage of sugar in there, and we're gonna go until the percent of sugar is equal on both sides. We'll get to that eventually. But this is what we're talking about with like this U-tube. This is what's happening. The percent of solute concentration will end up the same on both sides. That's what we're achieving. So water, if it was just the water that was going to balance, we get it to where there's 475 on each side, cool. Okay, if it's not the same concentration as sucrose on both sides though, more water's still gonna move until the sucrose concentration is the same as a percentage. Cool. So osmosis, diffusion of water down its concentration gradient across selectively permeable membrane. Can also be thought of as the diffusion of water from areas of low solute concentration to areas of high solute concentration. Right here we've already flipped the terms. We're telling you it's going from low solute concentration to high solute concentration. We flipped it, right? Because usually we're like, oh, high water concentration to low water concentration. Now we're talking about the solvent, or the solute. You will see it presented both ways. You will be given water concentration, you will be given solute concentration. You have to put into your mind what it is they are talking about. Picture it, diagram it if you need to. Something. It is going to trip you up. I am going to do the best I can so that you are comfortable with however the question is presented. I couldn't remember if I had something written down here. So please, if you're given any scenarios, situations, questions, are they describing the water concentration or are they describing the solute concentration? You've got to know.
Like yesterday, huh?
Yeah, I got it yesterday. See, it still feels.
Yeah, I could tell. It looks it.
Does it look brand new?
Yeah. I got this last year.
Did you?
Yeah. It says Merry Christmas on it. And I stole these socks from my sister.
Nice. I now have enough Christmas sweaters and sweatshirts to wear one every Friday.
That's a bit scary.
And then a couple other days, I still got t-shirts.
I have a vest.
I've got a cardigan sweater.
Oh. That's so much better.
Are we
Walmart.
You guys have a lot of.
I know. I'm gonna buy all of them. But I got it last year one. It was like the last wedding that they had. Like, sorry. I could get that. What time?
It's 10:13.
That's fine.
So your sister, is she dressed up? See? So I know like when I was in Saudi Arabia, yeah, it was like the red and white one they wore, and then the white one.
I'm so
It was
right.
just because make sure red and white one. Right, yeah, and the red and white one, you guys wear different colors for different seasons.
Gonna have to change my shirt. I'm wearing the wrong shirt.
No, it's just like the. I lost mine, I had it. I had red and white, yeah. I still have all
I
my.
just realized you said when Mexican people speak Spanish.
Who could from every country I visit.
That's not the Mr. White.
It's a little racist.
Oh, yeah, you guys are going
That's
10-15.
Mr. White.
Alright, another form of passive transport is facilitated diffusion. Diffusion of molecules through a membrane via transport proteins. Basically a channel in the membrane that allows molecules to diffuse across without the input of energy, so it's still facilitated diffusion. Increases the diffusion rate for things like water, small ions, right? Remember, water has a charge. It's polar. So if it's polar, can it diffuse directly across the membrane? No, remember, those are small non-polar molecules. Right? Water is polar. This is why, I don't know why we don't tell you guys this in like 9th grade and stuff when we talk about diffusion. Water needs the aquaporin. You too. Two categories of transport proteins, channel and carrier. Channel proteins are specific. Not just anything will go through the channel protein. Water can't go through any channel protein, it has to go through an aquaporin. Ions that have their specific proteins can't just go through a different ion channel. Cool, so specificity.
Right out there.
It happens without input of.
Thought it was gonna be cool today.
You guys still going? Alright. Why is facilitated diffusion passive transport and not active transport? Even though we're going through a channel,
scared to
why
be
is it
active.
still passive transport?
It's too scared to be active.
It's not
Because it's going to occur
spontaneously.
spontaneously without the input of energy,
Okay.
Oh.
right? It is going to happen without any outside influence, without the influence of energy or anything like that. Facilitated diffusion will still pass transport because substance proteins are helping move down, no energy is required. There you go.
Huh?
That's a lot.
It's still, you don't have to write it all down. What did I say? Yeah, no energy is required. That's it. That's the key concept to right here. It is going to happen without the input of energy. That's it. That's what you need from here, not this whole paragraph.
Can you repeat that again?
Yep, it's going to happen without the input of energy. So this is a channel. The channel, what type of protein is this? If I asked you to describe this little blue thing here, 2-word description, you would say it is a
Integral protein.
integral protein, right? Or transmembrane protein would work, right? Integral transmembrane protein, those are gonna be the channels for which we have diffusion occur and passive transport. What type of diffusion is this? It's going through a protein, what kind is it? Facilitated, absolutely. Even though it's facilitated transport going through a protein, is there any energy being used? No. You don't see a little star that says ATP, means no energy is being used, it's formed passive transport. The channel, hydrophilic. If it says it's a gated channel, can you just get through gates?
No.
No. You have to have some sort of ID or permission to get through the gate, correct? Right, that's why it's there for security. Same thing here. Has to be some sort of signal or something that can open or close the gate or to allow things to pass through the gate. Still not energy though. So only when the gate is open or stimulated will molecules pass through. And this is where I hope you get an appreciation of what biology is. This is where everyone's like, oh, biology is the easy science. Right? Remember, you're made of 73 trillion cells, give or take. Right? And this is happening, the processes we're talking about are happening hundreds or thousands of times per cell in your body, 24 hours a day, from almost the moment of conception until you die. All this junk has to happen over and over, and it just keeps happening. Right? This is life, dude. And the scale at which this happens, this is infinitesimally small for most of us, right? You're never going to see this happen. Right? This is why I think biology is much tougher. It is all make-believe. I can't show you any of this. That's the closest I'm going to get to showing you. Maybe as a video. Right? And the video is going to be somebody else's animation. At least in your other sciences, you can manipulate it. And you can watch it happen and things like that, right? Much tougher in bio. All conceptual. Aquaporins, these are the ones, like I told you, specific channel protein for water. Just think about the aqua gets to pour in. Huh? Aquaporins. Come on, dude, that's funny.
Wait,
So
what'd
the
you say?
easiest way to remember stuff is to make up stupid stuff like that.
Right? Yeah, there's no other way to do it. It's just, it's too daunting. There's too much out
So
there.
much.
Yeah. Oh. Yeah, 8, whatever. Yeah. Good. We squared away. Good. Moving on. Cool. This is what we mean. This would be a gated channel. So the channel sits like this until a molecule drops in. It will cause a conformational change. You will hear conformational change. And what that means when we're talking about enzymes or these, it's a change in shape. We talk about cell signaling, conformational change. Change in shape. So it's going to change its shape from this to this. and that's going to, it's gonna be here, this side opens, it allows it to dump in here, and once this is empty, this will open back up, bring another one, this side opens, boom, drops it in, that's it. It won't change this shape until the correct size and shape molecule lands in here. If it's a different molecule that wedges itself in there, it's not going to affect this the correct way. It's not going to open up and drop whatever that is into the cell. It has to be the correct molecule. So it is still semi-permeable. It is still selective as to what will or won't enter or exit the cell. Cool? Has to be the right fit for a conformational change. Carrier proteins undergo conformational changes for substances to pass. I wish I had remembered to say conformational change before I spoke. And once you get really used to the language or whatnot, you could sit at your next party talking about conformational changes and about anything, and everyone's gonna look at you like a freaking genius. They're gonna have no idea what you're talking about. Right? And you could be like, oh, look, the person was sitting on the couch, somebody sat on their lap. Oh, and it caused a conformational change. Look, they changed their shape because somebody sat on theirs. And everybody will know your best. I do it all the time.
Actually, I have that one smart uncle that actually did good in high school. So.
Yeah. Oh, you had one smart uncle that went to high school? Is that what you said?
No, that actually, I said that actually did good in high school.
Oh, I thought you said that actually went to high school.
No. Well, no, yeah.
So in terms of diffusion, describe what is most likely to happen to the particles in the image below. The dashed lines represent a plasma membrane. Does it look like those particles are permeable to the plasma membrane? It doesn't, but does that mean they can't pass through? No. So you can describe this two ways. You can describe it as if the dashed line was permeable to the particles, and you can describe it as if it was an aqueous solution where the particles were not able to pass through the membrane. You can describe it both ways. If we don't tell you what it is permeable to, it's open to interpretation. Active transport.
What? Where's that?
Huh? Yeah, we're skipping your practice problems right now. We're gonna come back to,
Oh, they're all practice problems.
yeah, there's a bunch. Cause you've got a lab in there that we're gonna run next week. There we go.
Wait, what do I call this lesson?
What do you call it?
For lesson.
Passive and active transport. So active transport, we spent all this time talking about passive transport, correct? Now we're gonna go to the active transport.
We've lost 2 minutes.
We only got 2 minutes left?
Yeah.
Damn it, Jim. Alright, let me see something. When I missed co-transport? Oh, I know what it was. Alright, so, where did we stop? No, I mean, before that. Alright, so, ladies and gentlemen,